Okay, so remember, I don't know what the last common ancestor for all mollusks looked like, but if you asked me to take a guess, I'd guess it maybe looked a little bit like this. This animal right here has the hallmark features that we see in mollusks, things like a ctenidium and a radula, which are uniquely molluscan. It also has this three-part body plan of a head, foot, and sensory organs, although they don't really show sensory organs here, but muscular organs. The visceral mass, which contains the coelom with the internal organs, and then this mantle that secretes a shell. So we've got this soft body, three-body part plan with a shell, yeah? And you'll notice it's got some more general characters. You'll notice, like most invertebrate animals, it has a dorsal circulatory system, it has ventral nerve cords, and it also has an open circulatory system, right? In the sense that it doesn't have blood contained in blood vessels. There's an exception for that in mollusks, and those are cephalopods, if you remember. Cephalopods have a closed circulatory system the way that vertebrates do and the way that annelids do, but most invertebrate animals have an open circulatory system. And you know a lot about this animal, right? You know that it's a bilaterian, you know that it has an open gut, right? There's both a mouth and an anus. You know that the blastopore gave rise to the mouth, right? Because it's a protostome animal. You know that it's a Lophotrochozoan, it's in that general clade. And like annelids and platyhelminths, it has determinate cleavage. Specifically, it has spiral determinate cleavage. That means that the cell plates are established upon the first division of the zygote. So you know a whole bunch of things about this guy, right? In addition to the fact that it's a mollusk, yeah? This shows you a veliger larva of a snail. You can kind of tell it's a snail, but it goes through a trochophore larva stage slightly before this, right? So that's, remember, one of the characteristics that puts it in this group of protostome animals. This picture is out of that invertebrate zoology book that I like a lot. It's intended to show this process of torsion. And this, as far as I know, is in all gastropods. So remember we're in the phylum Mollusca, right? And if we're in the class Gastropoda, right? This would be snails and slugs, basically. As far as I know, all gastropods undergo torsion during development. And basically what this does is it causes a helical coiling of the shell, but it's more than just that. It actually is a complete rearrangement of the internal anatomy of the snail, such that these little gills, which would be posterior, are actually brought to the front of the animal. Even the anus is brought to the front, so it kind of poops on its head, right? And you might remember that big group of gastropods that includes things like abalones and whelks and conchs and stuff are called prosobranch. That means what? Prosobranch. Branch refers to the gill or the ctenidium. Proso means in the front because it's brought to the front, yeah? And it stays in this final twisted pattern that you have following torsion. You'll see there's a group of sea slugs called nudibranchs that belong to this order Apistobranchs. Apisto means in the back, right? So they've moved their branch back to the back. You might say, well, why do they do the torsion? And then what they basically do developmentally is they do detorsion. They do it as they develop and then they undo it. That's what I'm saying. Every gastropod, as far as I know, does torsion, even though some undo it in the end and go through what's called detorsion. The torsion is so ingrained within gastropods and so fundamental that they all do it developmentally. And then, of course, we have a group that includes the land snails and slugs, which have a pneumostome. They don't have a ctenidium because, of course, they have to facilitate gas exchange between blood and air rather than between blood and water. And so they have like a little cavity that allows them to do that, a little moist cavity that allows them to do that. Okay, so phylum Mollusca. Classes that you need to know are Gastropoda, which are snails and slugs, right? Bivalvia, which are who? Bivalve means two shells, right? Clams, oysters, mussels, scallops, right? And then cephalopods, which you should know really well because I showed you a little movie on those. Who would those be?
Cuttlefish.
Cuttlefish, nautilus, squid, octopi, right? All of those guys would be down here. You'll notice there's another class on this little table out of Campbell, the Polyplecophorans, which are chitons. I don't think you have chitons in Florida. They're common in California and intertidals and stuff. I'll show you a picture of one. Polyplecophora means many shells. You'll see that they actually have 8 little shell plates. Yeah, but no real well-developed head. So I'll just show you a picture of those, but these are really the three that I need you to know. And of course, I'm going to break gastropods down into prosobranchs and apistobranchs and pulmonates, kind of the way that I was saying there. Okay, so this shows you a picture of a chitin. Have you all seen one of these before? It has this extension of the mantle around the edge called a girdle, and you'll see it has these 8 shell plates. This is sort of considered to be a more ancestral form for mollusks than gastropods would be. These guys if you were to pry it off of this rock, you'd see that it has a foot on it. It looks kind of like a snail, but with no head. And they actually make this little indentation in the rock where they sit. So they're really, really hard to pry off. It's not just like a snail that you pick up or something. They're really stuck into their little indentation. And of course they come out of it and they graze a little bit and then they go back to that little indentation of the rock and hang out there. The one kind of interesting thing I remember about these when I took like invertebrate zoology 100 years ago was that they actually have iron in their radula and they orient themselves to the Earth's magnetic field. And I can't remember whether there's some particular purpose for that, but I don't think so. But it's kind of an interesting little fact that you think, here's this little mollusk, on the intertidal, what's it doing? And here it is orienting itself to the Earth's magnetic field for some reason. Okay, so those I just want you to know they exist. It's another class within the phylum Mollusca. But here's the ones you really need to know. These are gastropods, right? This is the order, the subclass Prosobranchia. So these guys undergo torsion and they have their ctenidium up to the front, right? This is a conch, right? So when you go to Key West, you get key lime pie and conch fritters. That's the conch fritter part of that is this thing sliced up and battered and they fry it up. This, none of you seem to know in the lab. This is, abalone, right? Haliotis. Which this on the West Coast, of course, people go out and collect. It's considered a prized edible. This is a limpet, right? And this is a cowrie. And of course, there's a whole host of these. I mean, you saw some of the different shells that were on the table last week in the lab. If you walk into a shell shop around here, it's full of gastropod, prosobranch shells, right? Different marine snails. The one real hallmark feature I should point out, there are a few prosobranchs that are actually terrestrial. I had a friend that worked in Palau on a terrestrial group of prosobranchs. And then there are a few of the pulmonate, the lung-breathing ones, that are aquatic. So the way that you really know whether or not you've got a prosobranch or a prosobranch or a pulmonate snail is by whether or not it has an operculum. Prosobranchs always have an operculum and a pulmonate never does. So do you all know what an operculum is? It's the little trap door. You know how like when a snail goes inside of a shell, sometimes they'll have that little hard plate that they close behind them? That's the operculum. If they have an operculum, then it's a prosobranch. Okay. Here are the opistobranchs. These are called nudibranchs. What do you think that means? Naked. Naked. Branchs, naked gills. So here are their little gills, their little ctenidia, right, that are up in the back, and they're naked. And you can see that they're opisto in the sense that they are in the back. So these guys developmentally have undergone detorsion in order to get the little ctenidium posterior again. Brightly colored, really cool little slugs, right? Have you all seen these somewhere or seen pictures of them, hopefully? Usually most of them are kind of small. They just kind of fall along like little slugs. There are a lot of them that eat corals, right? So you all remember corals are in what phylum, what class? Yikes. Oh my goodness. How about Nigeria? And they're in the class flower animals with sea anemones. Anthozoa. Yikes. You guys really are all you're studying this, right? You don't just hear it like one time from me and they go, oh, that's good, I got that now. No, you're going home and drilling this somehow, right? Because you're going to need to know this really well. So they're cnidarians. What is the sort of hallmark character I said define that phylum that all cnidarians have, that you look for in Hydra, that Hydra has 4 different types?
Nematocysts.
Nematocysts, right? So here's something that's feeding on something that has these little cells, with triggers and stingers and venom, and some of these nudibranchs are actually able, believe it or not, when they're eating these corals, not only do they not get the nematocysts to fire, in other words, they take them when they're still mitocytes, when they're still in the cells without being triggered, they're able to manipulate them through their own body and actually put the unfired mitocytes up on themselves, like up on their little branchs, and use them defensively. How's that? Isn't that crazy? That they could actually steal the little mitocytes from the corals that they're eating and use them for their own defense. Isn't that remarkable? Okay, and here are the pulmonates, right? So things like garden snails and tree snails. That's a Hawaiian tree snail. And these are, what are these?
Slugs.
They're slugs. Specifically, they're banana slugs, right? Because they look kind of like bananas, and they're big. They're yellow. Sometimes people call them poor man's peaches because they kind of look like peaches. And this is the little pulmonate gastropod you should know better than any other, because this was my mascot. So back in the 80s, not long ago, when I was an undergraduate at the University of California, Santa Cruz, all of the students wanted the banana slug to be our mascot. And of course, the chancellor of the university wanted it to be a sea lion because he considered this to be not serious. But banana slugs are all over the Santa Cruz campus. If you ever go there, you see them all the time as you're walking between classes. And of course, the students there are a little counterculture, right? You realize it's mostly like surfers and hippies, right? You smell the pot as you walk across campus. The students all wanted this. The students won. So we're the fighting banana slugs. Of course, you guys are manatees, so manatees aren't horribly fierce either. So you probably don't think there's anything wrong with banana slugs. But back in the 80s, this was very controversial. The chancellor was very unhappy that he lost and that we became banana slugs instead of sea lions. Okay, bivalves, right? These are the guys with two shells. This is a oyster. This is a scallop. This is a mussel, like M-U-S-S-E-L, right? Not M-U-S-C-L-E, right? This is a clam. It's a razor clam, and this is a giant clam, right? These guys are all filter feeders, so you know, like in the cartoons where the giant clams grab people and chew them up, that's not right. You realize this thing just filters particles out of water. This right here, this thing that looks kind of like a feather, is its ctenidium, its gill. Remember that these guys have two posterior siphons, right? One incurrent, one excurrent. They bring water in through that incurrent siphon and move it over the gill, build up this mucus net that collects food particles, which is transferred to the mouth, and then of course water goes out the excurrent siphon, right? How about these little dots here? Any idea what those are on the scallop? They're eyes, yeah. They're little light-sensing organs. They obviously don't, they're not real sophisticated eyes, right? But they can sense light, right? So they do have little eyes. Remember, no head, no radula. They've lost those because of the highly specialized life that they live. You'll see on the practice exam, there's a picture just like this. Right? Very often I go through it. It's coming right out of the PowerPoint slides. I know one part of what the picture asks is to label dorsal, ventral, anterior, posterior. So you could all do that, right, with this animal. I think it asks you name the phylum, name the class, right? So look for this question. It's on the, I think it's like question 3 or something on the practice exam. So how about it? Here's the mouth, and here's the anus, right? So this is anterior, this is posterior. How about this would then be what? Dorsal, right? And you might kind of guess that because remember the circulatory system in most invertebrates is dorsal. These guys don't really have much in the way of a nervous system, but if they did, it would probably be ventral. This is ventral, right? Did you notice the coquina is kind of pointed on the anterior end so that it could go into the sand more easily? And you all know how it was doing that, right, with its foot. That it was basically extending the foot. How? By contracting the circular muscles, relaxing the longitudinal muscles, then it would make like a little anchor, right? A swelling at the base of the foot, and then it does what? It contracts the longitudinal, relaxes the circular, then it does it again, right? Contract the circular, relax the longitudinal, make a little anchor, contract the longitudinal, and relax the circular, right? And it keeps digging and pulling itself in, but it's kind of pointed on this end so it can do it. Notice that there's two adductor muscles that contract to keep the shell closed, right? This is the anterior adductor muscle. This is the posterior adductor muscle. No head, no radula, but it does have a foot, does have a mantle, does have a visceral mass. It has those basic features that we associate with a mollusk. And of course, this mantle is the part that's secreting the shell. And I think I mentioned on the practical, I almost always put a dissected squid and a dissected clam, and I could put a pin, like in the ctenidium, the mantle, and the arm of the squid, right? And then ask you, okay, I put a pin right here and say which part on the squid is homologous to the foot in the clam, and you would all say the ctenidium, the arm, or the mantle. the arm, right? The arms and the tentacles are basically the foot of the squid. The mantle, the part that you cut up to make the calamari rings, that would be homologous to this part. And the ctenidium, of course, would be homologous to the ctenidium in the clam. You guys are all going to get good with that. You realize when I asked this I used that question as a real question the first time around, and I asked anterior, posterior, dorsal, ventral, they didn't have a clue. To them it was just a clam, it was like round or something. Some of them I think even said it was radially symmetrical. Right? You guys aren't going to do that. You're going to learn this like for real, right? And then cephalopods, you know that I love these guys, right? I showed you a whole movie on these. Nautilus, cuttlefish, octopi, squid obviously would be in here. And these guys, if you were going to know something about them other than their kind of gross anatomy that you got from doing the dissection, they have a really well-developed nervous system, right? A spectacular camera-style eye, right? They're intelligent, and they have these pigmented cells in their skin called chromatophores that allow them to do all of these amazing color patterns in their skin and turn all these different colors. Yeah. Okay, we're good with mollusks? Now we're going to do ecdysozoa. So I'm only about a half hour behind. That's great. Okay, so to just kind of remind you really quickly where we're going. These are sort of most of the major animal phyla. The ones that we're treating are Porifera, Cnidaria, Ctenophora, and we've already done those. Platyhelminthes, mollusks, and annelids were your examples of Lophotrochozoans. I just finished mollusks right now, right? So what we've got to get through right now are the ecdysozoans. These are the guys we're doing in lab this week, and we're really focusing on two phyla in there, nematodes and arthropods. What does ecdysozoa mean?
Molting.
It means that they molt, right, or they shed their outer covering at some point. And then next week's lab focuses on deuterostomes, which is going to be echinoderms and chordates, and of course that's huge because chordates includes all of the vertebrate animals, right? And then the week after that we'll go to moat and kind of see everybody. Yeah. I'm hoping that you guys are starting to tie animals to names. I might as well give you a little heads up. You'll see on one of the questions on the practice exam, I give you a whole bunch of names, and then I try to get you to tie a biological structure or feature to each name. It works well as a practice exam question. If I gave it as a real exam question, it would be a bloodbath, I'm afraid, because I tried before and nobody understood it. They kind of understand it as we do it as a practice exam, but what I realized when I was doing it is that when I say a name, like if I say Scyphozoa, is there a picture in your head? Really, what's the picture? A jellyfish, yeah? That's good. If that's what you should have in your head. But what I realized is so many people have a hard time putting an actual picture in their head when they hear these names. That's more what I'm going to ask you. I'm going to give you 20 different names, and I'll tell you I've got like probably 300 names to pick from, right? I'm going to give you 20, and all I need you to do is give me a picture in your head. So if I said protostome, could you just name an animal? No, because jellyfish would be here. So you got to name somebody who's in here, right? So try again. Earthworm, great. Name another protostome. A clam, great. Or I say bilaterium. What could you say? A crab would work, right? Basically anybody who's in here. Just don't say jellyfish, comb jelly. How about if I say the hydrozoan? Can you name one? Hydra. You could say Physalia, Portuguese man-o-war, right? You could say Obelia. Y'all remember Obelia? How about if I said Turbularium? Can you name one? Dugassia would be good. How about Spanish dancer? You know those little flatworms that were penis fencing? Right? That works. How about if I said Trematoda? Could you name one? Clonorchus, the liver fluke, or you could say blood fluke. You can give me a common name. It doesn't have to be a scientific name. How about if you said schistosome? That's the little blood fluke that I showed you in that movie, right? You guys are going to get good with this, right? If I say oligochaete, can you name one? Elosoma would be great. Dara would be great. Tubifex would be great. Lumbricus would be great. Or you could just write earthworm, right? Yeah, are you all going to be able to do this? There's going to be a zillion more names starting right now because I'm going to start barreling through the arthropods, and you'll see arthropods are huge. You're going to have a whole bunch of insect orders to keep track of and subphyla and classes. It's going to be challenging. Somehow you got to put these names with a picture. You got to be able to tie a name to an animal. You're all going to work on this, right, with flashcards or something. Because seriously, 20 questions on the exam is going to be nothing but that. I'm going to give you a name. You got to tell me an animal that belongs in there. You know, the first semester I did this, I put mammalia, and they left it blank or got it wrong. Mammalia? I mean, how about like dog, cow, a mammal? Come on, guys, give me something here, right? Somebody put their name. That works. But I'm afraid that I'm going to lose you, that you get so lost in all of these names that you're going to get totally lost. You got to get not lost, right? You got to, like, a couple weeks here, you're starting to put it together, right? But I'm presuming when all these names are up here that you could somehow tie a picture to each one. Remember, this is more the phylogeny that we're really working with that shows sort of sponges as being paraphyletic, ctenophores and nigarians probably not being monophyletic. Remember placozoans, that little group that's potentially the sister group to the rest of the eumetazoa. This group of aseals that used to be platyhelminths but now appear to be the sister group to the bilaterians. And then this group of the other bilaterians here that we're dividing into deuterostomes and protostomes. We've already done this side of the protostome animals. Now we're going to do the other side, the ecdysozoa. I know I said that really fast, but I've said this like 10 times, right? You've seen this 10 times, right? So here are the protostomes that are ecdysozoans. Nematodes, roundworms, tardigrades or water bears, onychophrans or velvet worms. We are not treating these phyla. I will not ask you to name those specifically. Although if I said, give me a protostome or give me an ecdysozoan, and you said tardigrade or onychophran, that'd be great. I'd be happy if you said that, but you don't have to. You could just as easily say nematode, or you could say scorpion, right? Or spider, or crab, or butterfly, right? These are all arthropods, and you'll know them very well in just a moment. These are chelicerates, specifically arachnids. This is a crustacean, and this is another crustacean, because you'll see in a moment here that crustaceans, as historically defined, are not monophyletic unless you add insects. Insects, believe it or not, are crustaceans. Okay. This is a picture out of that invertebrate zoology book that I like so much. Remember that book was published in the 80s? This is what they thought in the 80s, and this is what they thought for a very long time, up until very recently. It shows a picture of this cute little polychaete annelid. This is probably who? If I say Polychaeta, who would you name? Nereus, right? So here's Nereus with his cute little, what do you call his little feet? Well, the CT are the bristles, right? But what are the little feet called? Parapodia, right? So his cute little parapodia with his little CT. He's got this nice little head with these cute little antennae, little eyes. Absolutely adorable. Here's an onychophran velvet worm. And here we have one of these upstairs for you to play with tonight, a millipede, which also has little segments, little appendages on each segment, a cute little head, little antennae. And so what, if you read this, the caption for this picture in the book is it talks about how annelids and arthropods are closely related to one another. You can imagine starting with something like an annelid. An onychophran could be the missing link. You realize we don't use that term anymore, right? But this was the idea that somehow this was sort of a transitional form that you could go through in order to bridge annelids and arthropods. And for a couple hundred years, this idea persisted that annelids and arthropods were closely related to one another, based largely on the fact that they have segmentation in their bodies. This has been all but completely disproven in the last few years. We now have a lot of evidence to support the idea that this little guy right here is much more closely related to a snail and a clam than he is to either one of those things. All the similarity that you see here is presumed now to be convergent. Segmentation evolved multiple times in different lineages, and all of that similarity here has absolutely nothing to do with common ancestry. Believe it or not. This is a Lophotrochozoan. These, which as are flatworms and mollusks, these are ecdysozoans. Yeah? This revolutionized biology a few years ago when they came to this idea. Most people couldn't deal with it at all in the beginning. Okay, so let's go through a couple of these, the two of the ecdysozoan phyla that we're going to treat. So nematodes, common name, roundworms.
Hopefully you realize the word worm doesn't mean anything, right? We talked about flatworms, segmented worms, and roundworms, and you'll see that there are ribbon worms and acorn worms and mealworms. Mealworms are beetle larvae, right? They don't have anything to do with all these other worms. All these things we call worms are just long and skinny, but they have nothing to do with one another at all. The word worm has no meaning in biology, other than as a general body shape. These are bilaterally symmetrical. Everybody's bilaterally symmetrical going forward, except echinoderms you'll see in adults are pentaradially symmetrical. But we're in the bilaterians here, right? So they're bilaterally symmetrical. They're triploblastic. What does that mean? You're getting good with this vocabulary, right?
3 germ layers.
It means they have 3 germ layers. They have mesoderm, right? They have idiosyncratic determinate cleavage. Remember we talked about indeterminate cleavage where the cell fates are not established on the first division of the zygote, and determinate cleavage types where they are, right? Where you can't have identical twins. Once the zygote divides, the cell fates are fixed. Campbell acknowledges at least 2 different kinds. If you went back to Campbell's 5th edition, it would tell you that nematodes and arthropods had spiral determinate cleavage. But once they started realizing that there were these different groups of protostome animals that were unrelated, they went back in and re-examined this. I am not a developmental biologist, so I couldn't begin to tell you what the difference between spiral and idiosyncratic cleavage is. And there's a paper in the lab supplements that suggests there's actually many more different types of derived determinate cleavage than just these 2. But Campbell, at least now in their book, acknowledges these 2 different kinds. Idiosyncratic, I think I told you, means weird. So even the developmental biologists didn't know how exactly to distinguish this from spiral. They just have spiral and then they have weird, right? But it's a determinate cleavage type that's associated presumably with this group of ecdysozoans. So arthropods, onychophorans, tardigrades, nematodes all have now this newly recognized type of determinate cleavage that's distinctive from the type that annelids and mollusks have. They have an open gut, so they have a mouth and an anus. They're protostomes, that means what? The blastopore becomes the mouth, and they have a pseudocoel. So what does that mean? They do have a secondary body cavity, but it's not completely lined with mesoderm, so it doesn't qualify as a true coelom the way that like what you'd find in an annelid. And where does a pseudocoel come from again? Doesn't come from a splitting of mesoderm or outpoppings of the archintron. Those both lead to true coeloms, right? This comes from the blastocele, from that initial cavity that's inside of the blastula. In other animals, it fills in entirely with mesoderm. In these guys, part of it actually becomes this secondary body cavity called a pseudocoel. The common name for these are roundworms. Interesting thing about nematodes, they have only longitudinal muscles. So you're going to see some little nematodes upstairs called C. elegans. Y'all know C. elegans? C. elegans is famous. And it's C. elegans, like this, right? I think some people thought I was saying C like S-E-E or something, right? The C is kind of like when we write E. coli. Why do you write E. coli? What is E. coli? Huh? Yeah, Escherichia or something coli. It's a bacterium, obviously, right? The E is an abbreviation because the genus name is gargantuan and hard to say, right? And hard to spell, so they just write E. coli. C. elegans is the same idea. This little guy is used for all different kinds of genetic and developmental biology studies. A little itty-bitty worm, you'll see him under a microscope. The genus name is something like Coranoabdites, and nobody wants to walk around saying that, so they just say C. elegans, right? But the C is just an abbreviation for the genus name. You'll see when you watch these little guys move, they look like little teeny tiny snakes, right? They propagate a retrograde wave, head to tail, right? And you'll see that they move in a serpentine fashion. You won't see any scrunchie. No getting short to fat, long and skinny, short to fat, long and skinny, because they don't have circular muscles. They're incapable of doing that. It's very distinctive. You can easily tell. You also see when you look at the cross sections through the Ascaris, the larger nematodes, you'll see that there's no circular muscles in there at all. You just see the longitudinal muscles, unlike the earthworm where you can see both. You also see in the Ascaris they have this tough cuticle on the outside that they molt, right? Kind of the way that an arthropod sheds its exoskeleton. And some of these are very important in genetic studies, specifically C. elegans, and studies of cell fate and cell type determination. I'll show you that in a second. There are a lot of them that are important pathogens. There's a whole society of nematodologists, people that study nematodes, that work with agricultural crops and other types of plants. They tend to be root pathogens in plants. There are some animal pathogens up in the lab this week. You'll see we have this beautiful dog heart with all of these heartworms coming out of it. Those are If your dog has ever had heartworms, those are a nematode infection. Do you know what elephantiasis is in people? You know what elephantiasis is, where people get these really swollen limbs and stuff? That's a nematode infection. So there are parasitic nematodes. Hookworms are nematodes. But there's a lot that are free living too. So there are nematodes everywhere. This is kind of an interesting group of animals in the sense that they are ubiquitous. They're found in almost every kind of environment, every kind of space you could imagine. And one of my instructors said, he was quoting someone else, that if all matter was to disappear, you'd have a perfect three-dimensional representation of everything in nematodes. If everything except nematodes disappeared, they're everywhere. In one little teaspoonful of dirt, you've got thousands of nematodes, right? You've got little nematodes on your eyelashes, probably on your skin, where you don't even realize they're there. If you pulled stuff out of your refrigerator and started looking at like your produce, you'd see little nematodes all over them. You have to put them under a microscope. Most of them are very tiny. When you look at the C. elegans up in the plate, you'll see that you can't see anything just with your eye, but when you put it under the microscope, you'll see that it's teeming with all of these little nematodes squiggling around in there. So there really are just nematodes everywhere. I went to a talk given by E.O. Wilson at Ecker College, and he used this statistic that if you picture all of the animals in the world, not species, but just individual animals, so how many people would there be in the world? Like, isn't there about 7 billion or something now? Six. Didn't we just cross the 7? I thought we crossed the 7.
We went back down.
Somewhere in there, a few billion, right? But now, like, try to picture, like, all of the earthworms, right? How about all the flies, all the fleas, all the aphids, all the mosquitoes, every little fish in the sea, every little shrimp, every little barnacle? Are you picturing every little animal in the entire world, every little clam sitting in the mud, every little type of insect you could possibly imagine, all the birds, everybody? Are you picturing how many animals are there? How many, like, numbers are there? Talking about billions and billions and billions. Obviously, humans are a relatively small-sized population compared to everything else, right? 4 out of every 5 individual animals on Earth is a nematode. Isn't that crazy? So every other kind of animal that you can imagine, every single individual other animal that you could possibly imagine, they constitute about 20% of the animals. All the other 80% are nematodes. These guys are absolutely, totally everywhere, and yet most of them are really, really tiny, and you never even notice them, and so you don't really realize that they're there or that they're as important. Think about all the little animals just in a drop of pond water, all the little rotifers and gastrotrix and everything else. Still, every other animal combined is outnumbered by nematodes 4 times over. Crazy, huh? You probably never even really thought about them until now. Most nematodes are separate sex. Guess who the one exception is? The one who's mostly hermaphroditic. Guess. Go ahead and guess. C. elegans. The one I'm going to show you is a poster child. The one I'm going to show you as the representative of a nematode. The only one that I'm going to have live for you to look at, of course, violates this critical rule. Most C. elegans, 99% of them are hermaphroditic, with like a tiny fraction that are males. There are no females, but there's a small number that are male only, and then the rest of them are all hermaphroditic. This is why, you know, that little schistosome worm that I showed you, remember the male and the female in the bloodstream, and they're producing all those eggs? I tend to think that that's a nematode, and it's because they're separate sexes. And you might remember that for platyhelminths, it's the other way around, right? Platyhelminths are virtually all what? Hermaphroditic, with the exception of the schistosome, which is separate sex. So you somehow have to get that into your head, that schistosomes are separate sexes, the rest of the platyhelminths are all hermaphroditic. Most nematodes are separate sexes. Like you'll see when you look at your Ascaris in cross-section up there, it'll have a male in cross-section and a female in cross-section right in the same slide. But C. elegans is hermaphroditic. There's C. elegans, isn't he cute? This drives home this idea of determinate cleavage. You notice you start with a zygote, right? One cell, and it's got to divide to produce a whole worm. There's several reasons that C. elegans was chosen to be a model organism, kind of like Drosophila or E. coli. One reason is it has a small number of chromosomes. It only has 6 pairs of chromosomes. The other reason is that if you have one of the hermaphroditic worms, get this. This is impossible for me to believe still, I've known this for a long time. It has exactly 959 cells in its body. Not 960, not 958.
It has 959 cells in its body when it reaches maturity. The male onlys have something like 1,032 or something like that. But again, it's an exact fixed number. So what they can do is they can map and figure out where every single one of those 959 cells came from. And you can do it almost like a little phylogeny here, right? Where you have the zygote. And notice on the first division, one of the two cells that comes out of the division is going to be this. The other cell that comes out of the division is going to be that. The cell fates are already fixed. You can't then pull them apart and each grow into a whole worm, right? One of the cells that's going to be this, that's programmed to be this, can only grow into that. You'd be missing all the other part of the worm. And the other cell is programmed to be this, and you'd be missing this part of the worm. Yeah? So this really drives home the idea of determinate cleavage, that upon that first division of the zygote, the cell fates are established. And believe it or not, they can map every single cell and figure out every single cell exactly where it came from. So most of what we know, because how many cells are in you when you're an adult? First off, it's not a fixed number, but it's roughly how many? A few more than 959, you realize, right? Like closer to 1 trillion. It would be really, really hard for me to figure out where every single one of your trillion cells came from, right? Much easier to map 959. Most of what we know about, you all know telomeres, the ends of chromosomes, and how they shorten, and they think that that's perhaps the mechanism for aging. Most of what we know about that actually comes from work with nematodes as well. There's super nematodes that have telomeres that don't shorten. They can live like 5, 6, 7 times longer than another nematode would. So a lot of what we understand about aging and how potentially to decrease the effects of aging comes from studying little worms. So I know these guys probably don't seem super important to you, but you'd be sort of amazed how much biology we understand by studying something like this. You could never get at those types of questions by studying humans. They're too complex. Okay, and these show you some little encysted nematode parasites inside of some muscle tissue. Any idea what kind of muscle tissue this is?
Pork.
It's pork, right? And these are little trichinella that are encysted. This is why you have to cook your pork really well. Because if you cook it really well, then you just eat these little guys. They're just a little extra meat. That's no big deal. But if you eat undercooked pork, you can get a disease called trichinosis that comes from this. So there's another nematode that's a parasite, right? Okay, let's do a couple questions. You notice I didn't have any questions in the mollusc and annelids because I was trying to get through it fast. Okay, so ecdysozoans, right? These are the molters, right? Are they protostomes? Do they have determinate cleavage? Do they include tardigrades and onychophorans? Or if you think ecdysozoans, if you think all three of these things are true for ecdysozoans, you can think E. Hopefully that's kind of easy. Do y'all know who are tardigrades again? Water bears, and onychophorans are velvet worms, right? Those are the common names for those two phylum. Okay, are we doing that, everybody? 13? There we go. Take a few more. Bunch more. Bunch more. How are we doing now? 19? Is that it? There's only 19 of you. I'm assuming that's everybody. Okay, well, if you pick those others, you're not wrong, right? But they're all true, right? Ecdysozoans are protostome. What's the other major group of protostome animals? Lophotrochozoans. Don't they have determinate cleavage? I just showed you determinate cleavage in a nematode. What does that mean? Cell fates are determined on the first division of the zygote, right? They have idiosyncratic determinate cleavage, right? Tardigrades and onychophorans. I just showed you a picture of them, right? Why do you think I showed you a picture of them? Because I'm talking about ecdysozoans. They're ecdysozoans right along with arthropods and nematodes, right? So all of those are right. Right? Okay, so which one of these doesn't molt? A grasshopper, a nematode, an earthworm? Or if you think all three of them molt, then you'd book D.
2 of them do molt. I guess maybe the other way I could ask you is, which one, if any of those 3, is not an ecdysozoan? Right? Because ecdysozoans all molt. Okay, we're ready? 3 more? That's easy, right? One more. Okay, good. Most of you got that. Earthworms are annelids. Annelids are lophotrophozoans. They're not ecdysozoans. Okay, so which one of these doesn't have circular muscles? You now know who everybody is, right? Nereus, C. elegans, Hydra. Who's that? Coquinus, right? The clam. So who doesn't have circular muscles? Who can't scrunch? Who can't get short, fat, long, skinny, short, fat, long, skinny? Okay, one more. Is there one more out there? It's you, Joel. The only person whose number I know. There you go. Okay, good. C. elegans is a nematode, right? Annelids have both circular and longitudinal muscles. Polyps, polyp nigarians have both circular and longitudinal muscles. And of course, bivalves do too, right? Didn't we talk about, how did I say that the coquinas dig, right? Contract the circular muscles, relax the longitudinal, make an anchor. Contract the longitudinal, relax the circular, right? They have both in their foot. Otherwise, they couldn't extend their foot like this and then scrunch it up like that. There'd be no way, right? So which animal is most closely related to a crayfish? You all know what a crayfish is? You will after lab tonight or tomorrow because you're going to dissect one. You all know what a crayfish is? It's like a little lobster. It looks like a lobster when it's about this big. Crawdad. What's another name for crayfish?
Oh, yeah.
Yeah. Well, crawdads I think are actually a little bit smaller. Basically, it looks like a lobster, only it's about this big and it lives in fresh water. Do y'all know what a crayfish is?
No,
So the question is, what's it most closely related to? A nematode, a butterfly, an earthworm, or a snail? Now, if you know what you're doing, you could get this into a phylum. These are in 4 different phyla. Only one of them is in the same phylum as that one. So if you know what phylum each one is in, this is easy. Okay, 2 more. A crayfish is not a fish, you realize, right?
Oh.
Honestly, if it was, I don't know what the answer would be. Okay, are we there? One more? It's not Joel this time. Is there one more? Check your number. Okay, there we go. Okay, crayfish. Yikes. Crayfish is an arthropod. This is a nematode, right? Phylum Nematoda, right? Butterflies are insects, they're arthropods. This is an annelid, and this is a mollusk. Didn't I just do this a minute ago? Right? You guys are going to learn these names, right? You're going to stress me out if you don't learn these names.
like the file at least. Okay, so here we go with arthropods.